It is an honor and a privilege for me to be here, and for those of you who I have met before, Alex's Lemonade Stand is the reason that the Phase 1 program exists in many ways at Children's Hospital. So I will say that very clearly. So I have a very long-standing investment and love of this organization because when I wanted to start the program at Children's Hospital, I needed money to pay for a research nurse and a data manager and some startup, basically, that is impossible to get from traditional granting mechanisms. So the two organizations that I feel are responsible for any success I have had are the Morgan Adams Foundation and Alex's Lemonade Stand. So thank you for being in this room tonight. I thought if I went from the landscape of Colorado to the landscape of the atrium at Children's Hospital, that gives you a pretty decent perspective about what we talk about. What I wanted to show really, and some of these lines show up better than others, the model for success in oncology overall is childhood cancer. And within childhood cancer, the model for success in childhood cancer therapy is the single most common type of cancer in children, which is called acute lymphoblastic leukemia. About a third of children are affected by ALL, and across the country, there are about 16,000 kids a year diagnosed with cancer. That compares to 1.6 million adults a year who are diagnosed with cancer. So proportionally, pediatric cancer is a very small percent of the pie, but what we lack in numbers, we certainly make up for in power. And I think one of the things that we have been able to do is constructively collaborate with colleagues around the country and around the world to successively improve the outcomes for kids with cancer. And so I want to show this simply because in acute lymphoblastic leukemia, if you were diagnosed in 1968 to 1970, you had a less than 10% chance of surviving. And in fact, the first known survivor of childhood ALL is actually now a 60-something-year-old elementary school teacher who lives in Pennsylvania. And she was diagnosed in 1968 when the odds were really pretty poor that somebody would survive this disease. And every couple of years, every sort of generation of clinical trials that we've been able to do for childhood cancer, we've been able to improve the outcome in ALL. And that's not because we've introduced a lot of new medicines or a lot of great therapy. It's because we've optimized the therapy that we have. So the drugs that we used in 1968, we still use today. We've added a few more in there. But this curve up at 2006 to 2009, those of you who have been at Children's will know that that trial was actually led by Dr. Kelly Maloney, the largest childhood ALL study done in the world to date, almost 6,000 patients. And in that trial, there's a subset of kids that has a 99.4% chance of being alive. And that's 10-ish years later now, more or less. So that has come simply because we talk to each other and we collaborate with each other. This is not because we've gotten a lot smarter. It's not because we've gotten better at talking to each other and better at designing clinical trials that get us to these incremental improvements. So I'm gonna start with a patient story. This is a patient that none of you knows, but she is one of these girls, and they don't show up very well, I'm so sorry, because they're all adorable, so that doesn't matter. But one of these girls actually came to us, and I saw this family. They relocated from Northern California, from the San Francisco Bay Area, because 3 different hospitals there told their parents that there was nothing they could do for this young girl anymore, that her leukemia had come back. It was very aggressive to start out with. It had come back very, very quickly within 3 months of a bone marrow transplant, and there were really no viable alternatives for her. So they came to us because they found a clinical trial that was one of the first we did at Children's at the old hospital, and said, we don't have anything to lose. We would love to try something that you can offer for us because we know what the outcome is going to be otherwise. That was on the 4th of July. So we're coming up on an anniversary for this family. I met them in clinic on the 4th of July, and on the 5th of July, we started her on this very experimental drug at the time that had never been used in a young child. It had only been used in older adults. And 2 weeks later, she was in remission. And 2 weeks after that, she was still in remission. And 2 months after that, she was in remission. We had to sort of figure out what to do with her because we didn't really expect that. It was pretty great. Nobody was complaining. But that is the mystery of what we do sometimes, is we don't know how an individual child's story is going to evolve. And after about 8 months, the family was trying to figure out how they were going to continue to have some of them in San Francisco and some of them in Denver because this was an experimental drug. It was very highly regulated by the FDA. They couldn't just go off and get it even though they have major medical centers in San Francisco. And after about 8 months, we decided to stop the treatment because we didn't know what else to do. And that was in 2013. So at the end of the talk, I'm going to ask you to guess which one of these kids was the patient. So I talked a little bit about how we haven't gotten dramatically smarter, but we've gotten better at working together in how we treat childhood cancer. And our mode of treatment hasn't really changed that much over the years. So we still have the really difficult period of time when a patient is diagnosed. We try to get them into a remission quickly. That usually is at least through some initial chemotherapy, if not a combination of chemotherapy and surgery or radiation. And then we try to do what we call consolidate that remission. So we give more intensive therapy. Sometimes we give therapy that's directed at the central nervous system, or patient has surgery that's really a definitive surgery to try to localize their disease. It might include radiation therapy in some aspect. We oftentimes will give additional chemotherapy after these moves. And then for some patients, maintenance chemotherapy is important. And maintenance chemotherapy is not used in every type of disease, but it often can provide a constant level of pretty generally low dose, very well tolerated therapy that serves to really lock in that remission. And we learned, for instance, in childhood acute leukemia, for ALL, if we don't give maintenance, kids will relapse faster. So this is a very important, although relatively low intensity, not terribly exciting period of therapy, but it's really important. And all of those clinical trials that have helped us improve survival have helped us optimize each of these stages. I kind of made this as broad as I could, so every different kind of therapy has different elements to this. But we've learned over those trials that if you eliminate any one of these blocks, you're not going to have as good an outcome. And as exhausting as it is to be a patient or a parent going through this, each one of these is really important. And I leave maintenance in there because in all likelihood, you have seen today or will tomorrow or did last week a kid who's getting maintenance chemotherapy and don't know it because they look exactly normal. They look just like any other kid on the street or any other kid in your school. The statistic that I like to quote is that 1 in 200 kids is now a survivor of childhood cancer. So if you think about your school, your kid's school, the community that you're part of, whatever that is, there's probably a kid who's a cancer survivor. Many of them look absolutely just like every other kid, even in the middle of therapy sometimes. So it's important to sort of think about that as we evolve our concept of what childhood cancer is and does. So I talked a little bit about how we haven't gotten a lot smarter about our therapy. We use really old drugs. This is treatment for ALL. And when we look at the drugs that are most common, these are drugs that have been around a very long time. They're older than not only our patients, but many of our physicians. Fortunately, most of them are still older than I am. But what's really remarkable to me is that there has been very little advancement in ALL therapy over many years. And in fact, this drug that was approved in 1990, nobody ever uses for ALL. It is absolutely not part of our normal therapy, but it does have an FDA approval for ALL. But if we go from 1979, which was a very important drug for us to use in leukemia therapy, it took us all the way to 2004 to approve another drug for childhood leukemia. And in fact, this isn't even a commonly used drug. It's only used under very unusual circumstances and for very few kids. And then it took us another 10 years to get the next drug approved. So for somebody like me who has spent most of her professional career trying to get drugs approved for childhood cancer, this is not good news. And it's relatively frustrating, but what's really exciting about this is that this drug at the end, 2014 was its first approval, 2016 it was approved for children specifically, which is really exciting. That drug was approved on the basis of a trial that we led at Children's Hospital Colorado, and that was probably to date, I should probably just give up because I'm probably never going to get another drug approved in my life. But it's really exciting because it means that the kids that we had the opportunity to take care of were part of this. This was 20 institutions around the country, 10 in North America and 10 in Europe. And we moved the needle, which is really exciting. I'm going to talk about that in a little bit. But that 10-year gap is too long. And so that's one of the things that we're working really hard at. So I'm going to make some assumptions. I'm going to assume that we should continue to try to improve the outcome for kids with cancer. So even though there are a lot of kids that do very, very well, there are kids with 90%, 90+% chance of outcome, That's not true for every kid. And so I don't think most of us are going to rest until that is true for every kid and we do even better. We do know that there are still some kids that don't do as well, that have a poorer prognosis, or they have a rougher time, or they require much more intensive, much more toxic therapy to try to get to a cure. And so one of the questions that we have to think about is how can we get there? Can we do better? What is it going to take to get better? And we also know that many of our therapies, which a lot of you know firsthand, while they are effective, are very toxic. And they're toxic short-term and they're toxic long-term. So what I would love to do is to be able to do better with that. How do we specify exactly the right combination of therapy for exactly the intensity that we need, the duration of therapy that we need to give? And how can we limit some of the toxicities that we know are compromising to people's outcomes and their long-term ability to have a high-quality life? One of the things that we've tried to do then is to bring forward these new drugs that everybody hears about now. They're on the cover of Time magazine, they're in the New York Times, they're everywhere. And what we want to do is make sure that we can figure out how to use them appropriately. So I'm going to talk a little bit about that. But one of the frustrations has been the ability to introduce these really new and exciting drugs, especially to children early on in their development. There are lots of good reasons that we don't use children, as one of my family said, as test monkeys. But we need to make sure that we know what we're doing when we give a new drug to kids, not only because we may not know what its side effects and consequences are, but we also might not know what the long-term consequences are. So one of the things that we learned over time is that with a particular class of drugs, it's very effective against several particular kinds of cancer, but it actually can stop growth. And so I would venture to bet that every family I've met has said, I would be okay if my child was a little bit short. I want them to be alive. But what if we didn't have to make them short? What if we could also help them attain normal growth? And is that growth inhibition a normal effect of the drug that we need for the drug to be effective? Or is it just sort of a pesky side effect that we should try to do better? And those are the things that plague us every day when we're trying to think about clinical trials, so that we understand what those side effects are and we understand who's most at risk and who can we prevent that in. The next thing is kind of, again, as I said, it's a problem of privilege in that many kids are cured today. Every year on average, there are about 950 new drugs that have entered clinical trials for oncology care. 950 different drugs. That's almost as many kids who relapse a year in the United States with various diseases. So you can't just dole out one drug to each individual child and figure out if it works. It's very hard to think about how many of those drugs is actually worth testing in a pediatric population. How many of them might be useful and effective? They might be drugs that are developed for adult cancers, but they still could be effective for pediatric cancers. And how do we choose? So that's an important thing for us to understand. The next set of things or challenges that we think a lot about is that as our technology has improved, our ability to diagnose cancer at the molecular level, and that was the phone call I was taking earlier, at the very gene-specific level for an individual patient has really evolved. It's exploded, in fact. So what we are able to now do is take an individual patient's cancer, put it through a barrage of tests in the lab, in a computer lab, in fact, after it goes through the regular lab, and come up with what I've talked to for many people about basically a molecular fingerprint. What is the individual profile of this patient's tumor? And what do we know about that profile that we can match up with appropriate therapy? Or there might be some really exciting new drug that we can fit specifically to match up so that we know that we're treating the thing that's abnormal in that tumor and hopefully leaving the healthy tissue alone, leaving the healthy growing individual child alone. So the molecular profiling component of this has meant that we don't just have one type of diagnosis. We don't just have a sarcoma. We don't just have a bone tumor, but we have sub-diagnoses and sub-diagnoses and sub-diagnoses. So we might have medulloblastoma, and among medulloblastoma, we might have MYC-driven medulloblastoma, or group 3 or group 4 medulloblastoma, and among those, they have even more individual profiles. We might have ALL, but I'm going to show you on the next slide, we have a whole bunch of different kinds of ALL. And those change not only among individual diseases, but even within an individual population. And how we look at those sub-molecular subtypes makes it even more challenging for us to figure out how to treat them, because each of those gene abnormalities might confer a different response to different therapy, and we need to be aware of that so that we are only treating patients with one particular gene abnormality with a particular drug that affects that pathway but doesn't affect another pathway. I'm going to save a little bit about the regulatory processes that control drug development because I spend a lot of time thinking about it. I was at the FDA last week and I'm going back tomorrow to talk about regulation in drug development and what happens, why don't kids get sooner and quicker access to clinical trials and promising new agents. But part of it is that on average today it costs about 1.2 to 1.3 billion with a B dollars to develop a new cancer drug. So if we say that there are 30 kids a year that might benefit from that drug, the math is pretty simple and most pharmaceutical companies aren't going to be willing to do that math. So it's our job to try to figure out how to broaden the applicability, how to think about how to not only help that drug be successful if it's a good one, but to make people interested in developing a drug that's probably not very financially beneficial for them. It's a very long political discussion, which I will refrain from. But I do want to talk about this because I think this is actually really important and I don't know how well this projects. But even if we have the same disease, that disease varies by age and by risk factors that we've been able to develop over time. So along the bottom there is childhood standard risk ALL, which is kids between the ages of 1 and 10 at diagnosis, with very specific parameters about how high their white blood cell count is and some other biology. The next chart over is same age, 1 to 10 years of age, but slightly higher risk biologic features at diagnosis. The next one over is teenagers, and the next one over is young adults, so 18 to 25-year-olds. Remember, this is still called ALL. This is the most common type of childhood cancer. And then each of these colors represents a different, relatively well-defined molecular subtype of ALL. So you can have ERG ALL, you can have hypodiploid ALL, you can have MLL rearranged ALL, you can have a whole bunch of different kinds of ALL, but their definite incidence, predominance, and risk, ultimate good, bad outcome, whatever their prognosis is very dependent even on age. So we know, for instance, that the green box at the far left, which is that bottom 25 to 30%, is one of the most common types of ALL in children, and it's also one with the best risk, right? I mean, those are the kids that have a 99% chance of being cured. So it's great if a third of our kids with ALL have this, but if you're an adult, it almost doesn't exist. And so not only do we feel somewhat comfortable that we can cure a lot of kids with ALL, this is exactly why adults don't think ALL is such a great disease, whereas we're generally feeling pretty optimistic about it because they have fewer cases of the best risk ALL and they have more cases of the highest risk ALL if you're an adult. So these things vary, and this is why having this molecular sort of fingerprinting is so important for us to understand how best to treat the patient that's sitting in front of us, not just all statistics and all numbers. So what are the next assumptions that we're going to make? The FDA, you'll probably, it feels like every day in the last few months, there's been a new drug approved for some kind of cancer. I get an email every day. Pembrolizumab approved for third-line non-small cell lung cancer, which is great because more drugs are out there, there's more competition, there's more development, there's more availability. But it also challenges us to try to figure out what do we do with these, because most of the time these new drugs are developed in patients who've already had standard therapy. And we don't know what happens if we move those very exciting, promising new drugs really to the front of the line. It's also a little bit challenging because if we sat down and I said, you know, you have a 90% chance of being cured of your disease, but we'd like you to enroll in a clinical trial and we really don't know what's going to happen. That's a pretty hard thing to sell. You might be willing to do that for yourself, but you're certainly not going to be willing to do that for your child. So it's important for us to think about, as we're developing clinical trials, what is the real risk? What is the benefit or the potential benefit? And how sure of ourselves can we be as we have those discussions? The next thing is to really underscore, and again, it's a little bit, I'm a little convicted in my beliefs about this. I think we should do clinical trials. I think clinical trials are the way that we've advanced therapy, that we've helped cure kids today. And unless we all share that information and collect the data that help us know that we're actually making really good decisions overall, we're not going to learn anything. We're going to be destined to repeat mistakes of the past without gaining anything from them. And I think it's really important, as I sit in front of a family and propose a clinical trial for somebody or a particular therapy, I want that family to know that I'm not only taking their kid's life very personally and that it's important for them to understand that. But their child's life actually matters not only for them, but for the greater context, that they are actually contributing to what we're going to do to make those curves get better and better and better in the future. And we talked about the problem. There are 950, 1,000 new drugs. We can't test all of them. We can't even test a fraction of them every year. So we've actually joked that there are more drugs than there are patients to treat, which in some regards is true. And that makes us need to be more responsible in how we design our clinical trials and how we think about using these drugs for kids. So what do we do now? On the left, we've talked about sort of all of these dilemmas and all these challenges that we have. So one solution is to say, we do pretty well. Most kids are, doing okay, and we're not going to do anything different. There are some groups of patients that we can't do a lot better with. I told you that there was a subset of patients that has a 99% chance of cure. We can't do a clinical trial to go between 99 and 100%. They're statistically not possible in any of our lifetimes. It would take probably 30 years and 20,000 patients to be able to do that. I'm not a statistician, which I've just proven. But it's important to recognize that we have to focus on the kids that we really can do some significant good for and make some significant improvements for. So I'm not happy with that answer. But I do think that there's some things that we can do, and one is to add some of these exciting new therapies in an appropriate way to change how we're targeting cancer cells overall. We understand a lot more about cancer biology than we did in the 1950s and 60s when many of those drugs were first used. We should take advantage of that. We should really build on the experience of that. And because we have this depth of understanding, it allows us to be smarter about how we design our treatments. The next thing we can do is try to take some of these new drugs and swap out the old ones that have more toxicities and more side effects than we'd rather have. So we had a discussion today. One of the relatively common cancer therapies for most leukemias and several different solid tumors is a really good drug, but it has long-term effects on the heart. It makes the heart muscle squeeze less well. There's a new version of that, which is a little bit of a designer drug, but it essentially removes all of those cardiac side effects. It's just as effective. But what if I said, we're going to give you a drug and you have a 20% chance of having long-term heart disease and heart failure because of it, and that might not show up for 20 years, but you might be on heart meds for the rest of your life. Or we have the same opportunity to treat your cancer effectively, and there's no cardiac risk. Pretty easy to choose which one you'd prefer. Getting that drug into kids is a little bit more challenging from that. But if we can substitute out these drugs that we know are effective with better versions of themselves, that's going to be a tremendous advantage. The next thing to think about is, are there some places where we can actually reduce therapy? And a lot of the work around the world that we're collaborating on right now is to try to do what we call therapy reduction. There are some patients who we probably overtreat, who may not need the volume and the intensity of the therapy that they're getting, until we ask the question about whether or not we can take away some of that therapy in a very organized, very thoughtful way, we won't know. There was a very well-done study conducted by the German cooperative group that actually looked at shortening maintenance. I just showed you about how maintenance chemotherapy is important for some patients. They tried to just reduce the amount of maintenance that was given, and it didn't work. They actually had more relapses on that arm that reduced maintenance. We wouldn't have known that if we hadn't done that study, and that's really, really important to know. The good news for the patients on that study is that they could go back into remission, so it's important for you to hear that too. And then the next thing that's really important, and many of the consults that I get are patients who are the high-risk patients. They're the patients whose cancer has come back. They're the patients whose cancer might have come back several times. I think the patient that I've taken care of with the most recurrences right now is on recurrence number 9. That gets old when you're that patient, when you're that family. It would be great if we could do better for those patients so they don't have to have recurrence number 9. So these are the things that we're going to focus on, and I'm going to talk about how we can try to narrow that gap to get more kids cured at less expense to them in terms of their quality of life, the outcomes for them long term. So we know that we have these smaller and smaller subsets of disease. We have smaller pools of patients to study in clinical trials. So what does that mean we do? We have to do better clinical trials. We have to cross borders. We have to cross country bounds. We have to go across oceans to be able to collaborate on this data. And that's one of the things that I love about my job, is that on any given day I have a telephone conference that goes across 8 or 9 time zones if I'm lucky, and sometimes more, because somebody's always in their jammies at that point. But so today, for instance, I talked to France and I talked to Australia, which is really funny because they're completely And we're kind of stuck in the middle, but that's a fun telephone conference to be on because it really allows us to say, how can we do this better? What are the things that we can do? And everybody wants to make this better, which is great. It just means that we have to be creative in our scheduling. The next thing that we think about is how can we get these exciting new drugs into kids? How can we do it sooner? There will be a paper that will be published this year that effectively has been agreed to by the FDA, which I'm really excited about, that says that kids who are 12 and above can get access to these drugs. You don't have to be 18. But what it means is that if there is good biologic reason for a 12 or 13 or, heaven forbid, a 17-and-a-half-year-old who has a disease that might be really amenable to treatment, and we say, no, you can't get this drug because you don't turn 18 for 3 months, there's no biologic reason for that. And we're doing much, much better working both with the pharmaceutical companies and the FDA to really harmonize those agreements about how quickly kids can get access to these drugs when they need them the most. So I'm very excited about that. The next thing that we talked about is how can we sort of limit the intensity that we've gotten to for therapy, add new drugs, substitute new drugs, swap out old drugs for new drugs. How can we make those better? So these are the things that are actually achievable. So there are a few things to think about, and some of these you already know, so I don't want to spend a lot of time. But when we think about how we design cancer therapy, these are things that are really critical. One is, what's the disease we're treating overall? How much tumor is there in the body? What is the total body tumor burden? Where is it? And how quickly does it spread or divide? How quickly do we need to take action? Part of that also is how quickly a tumor divides depends on also how susceptible it's going to be to different therapies that we give. And when we give those therapies in the kind of life cycle of the tumor really depends on ultimately how successful those therapies are going to be. It's also really important as we're testing new drugs, as we're evaluating new drugs in clinical trial, to understand what effects on the body is the disease responsible for versus what effects on the body is the drug responsible for. Sometimes that's really hard to separate. And it can be a challenge to try to weed and tease those things apart so that we try to figure out what is the disease itself versus what is our treatment of the disease in terms of causing side effects. The next question that's really important to ask is the central nervous system relevant? And my colleagues who are neuro-oncologists will say, well, of course it's relevant. But the nervous system is a pretty cool thing. The nervous system is there for a really good reason. But the central nervous system is also one of our biggest enemies in cancer therapy because the body has this thing called the blood-brain barrier. The blood-brain barrier is there for a really good reason. If we cut ourselves and get a little bacteria in our finger, the blood-brain barrier keeps that bacteria from floating up to our brains and causing meningitis or abscess and all kinds of other evils. But what happens then is if we give chemotherapy in our circulation, it doesn't always get into the brain. And if the brain is either the place where the tumor is in the first place, or it's a place where it could potentially metastasize, tumor cells hide out in there, and we have to treat the central nervous system differently. So one of the questions is, how do we design a drug to get into the brain just when we want it to, just at the right time? How do we know which drugs actually even get into the central nervous system and appropriately, or if they get in and maybe they cause too much toxicity, how do we think about that? Those are all really relevant things to think about in terms of how we adequately treat and how do we appropriately treat. And then the next thing that I've done this in a very reductionist sort of way. One of the important questions that I think about when I design a clinical trial is, how fast can this disease hurt somebody? How much time do we have to deal with? Is this a very aggressive disease that could kill a patient in a matter of a few days or a week? Or is this a slowly progressing disease, and if the drug doesn't work that fast, we can afford to wait? So those are important questions to ask, because when we start combining drugs, we need to know what our window of time and opportunity is to make sure that we're treating a patient before the disease gets them in trouble. And then, one of the questions that's important to ask is, how treatable is this in the first place, and at what cost? So there are a lot of kids today who are alive and well, but at a pretty high cost for themselves and for their families and for the communities that they're part of. And one of the questions is, can we do better with that? That's an important question to ask, both short-term and long-term. And it's something to always remind ourselves of as we're thinking about this, so that we are doing right by the kid that's in front of us, and that's an important thing to do. So what are clinical trials? What's available? This is kind of my way of thinking about a clinical trial. If I'm assessing a trial and somebody says this is an opportunity to participate in a clinical trial, what does that really mean? So I want people to ask who's running the trial and who's going to benefit from it. That's pretty simple. what's the reason you're doing this trial? What is the fundamental question you're asking, and why do you think this is a good idea? That sounds a little bit funny, but where are the data that help us think that this is a good idea? That's a very appropriate question to ask. Has it ever been done in people before? Has it ever been done in kids before? Has it worked? And what are the risks? It is amazing how often we have had the opportunity to do first-in-human trials, and that's a pretty sleep-losing endeavor for those of us who are responsible for them. But it's also very exciting. And it's very exciting because we think we can do better. And we would never do a trial without thinking that we can do better, or just saying, oh, it might be interesting, let's try it. Trial design is a whole entity in and of itself, and we won't spend too much time on that. But one of the things that I always encourage people to ask is this a randomized trial? Do you flip a coin and decide which therapy you're going to get? Is there only one arm of the treatment? Or is it a placebo trial? My own bias is that placebo trials really have no place in oncology therapy. There might be one sort of circumstance in which that's an option, and that's if there is no therapy available. If a reasonable alternative is no therapy, then a placebo trial would be okay if you're willing to have no therapy. Other than that, we don't do placebo trials. Not part of our portfolio, not part of our sort of psyche around that. What are the alternatives? One of the things that would be important to ask is, what would you do if I weren't on the trial? What would my therapy look like? Would it be the same or different? Would it be similar? Where are the sort of the fine points and where are the questions? And then who's taking care of my kid that you've just told me you have to treat? You want somebody that does this a lot. You want somebody that has an enormous amount of experience. And while every kid is individual, you want the person who's taking care of your kid to have done this a lot and to not do it just a couple days a week occasionally. You want this to be their life, breathing, living every moment of what they do. And you want their friends and colleagues to be exactly those same people at other institutions, so that if you do come up with something that isn't perfectly clear, you phone a friend, and that friend is willing to pick up the phone at 2 in the morning. We do that for each other, and that's really, really important. What about the drugs that we're talking about? How do they work? Are the side effects ones we expect? Are they unknown? Are they short-term? Are they long-term? Does it get into the brain? Should it get into the brain? What if it does get in the brain and it's not supposed to? What are the data that have led to the development of these drugs? How do we think about them in some sort of construct so that we understand what we're doing? And what we call on-target effects. So if we take a pathway and we say, this enzyme is really important for cancer cell growth and division. Well, cancer cell growth and division is the enemy, right? That's what causes cancer to get worse. If you stop that enzyme, if you shut it off and it shuts off cancer cell growth and development, that's great. What if it shuts off normal cell growth and development? And what kind of cells does it shut off? So those are the kinds of things that we need to know. I'm going to give you an example in a little bit about a type of therapy that actually requires people to get pretty sick for a short period of time for it to work, because it's stimulating the immune system to fight the cancer. If we don't trigger the immune system and you don't get those flu-like symptoms, you don't get a fever, your blood pressure doesn't get a little soft and wonky, the drug's not working. And that's a very different way of thinking about side effects. I actually have patients on a couple of these trials, and the first thing the family says is, are they sick enough? Because they know they have to get sick to get better. And that's really a weird way of thinking about it. But if you see the side effect, is that the side effect you're hoping for? Or is it sort of a byproduct of the drug that you'd really rather do without? And can we make designer drugs to breed in the good ones and try to breed out the bad drugs or the bad side effects? And then, another question is, if we're going to start a clinical trial, for instance, we're going to treat with a new drug. Have adults gotten this drug before? Have kids gotten it before? Have they gotten it for the same disease or a different disease? There are a lot of drugs right now that are being used in a variety of different cancers all at the same time. It used to be that if you had lung cancer, you would only get this particular combination of drugs. And if you had colon cancer, you'd never see those drugs. You'd see a different group. Now we're learning that there are different molecular subtypes of what seem like different cancers on the surface that actually have the same biologic driver. And therefore, the same drug can be very effective in each of those. But it doesn't intuitively make sense based on how we've treated cancer today. So I'm going to talk very briefly about just a couple things that I think are really new and cool and interesting. And I'm going to talk a little bit about sort of what's happening in childhood cancer overall. One is disease-directed teams. We have gotten more and more and more subspecialized. We joke that you used to have a bone tumor, but now you have a left upper shoulder tumor. And the surgeon can't operate on the left side, he can only operate on the right side. And it's not quite that bad, but we have gotten much more specialized. So even within leukemia, most of my patients are not patients that are newly diagnosed. I don't see patients when they walk in the street, unless I happen to be on call that night or that weekend. My patients are patients that have a separate subset of disease. And many of my colleagues are the same way. They're neuroblastoma experts, but they're neuroblastoma experts that have a particular interest or a particular focus. They're bone marrow transplanters that only do a particular kind of transplant, for instance, or that's their particular area of specialty. And so we have a lot of subspecialty teams. Most of them are very multidisciplinary in nature, because that's what we need to do to talk to each other. But the more our knowledge and our technology have evolved, the more our specialty has evolved as well. Genomic profiling, I talked about that thumbprinting, and there are increasing numbers of patients who are actually having their tumors profiled, and we know what their tumor looks like, not only at the microscopic level, at the submicroscopic level, but really at the molecular, the DNA, and sometimes even the single cell level. There is an increasing, I think, very appropriate emphasis on what we call supportive care or wellness. Psychologists, social workers, child life workers, art therapists, physical therapists, occupational therapists, all of those people are critical to kids having a good outcome for what they're being treated for. And we're also introducing a concept earlier in therapy, which has shifted a lot. It used to be that when you said call the palliative care team, that was actually equivalent to calling hospice care. And in some institutions, that's probably still the same part. But palliative care is actually a whole separate entity, and it involves things like pain management, optimizing kids who are at home, quality of life, rather than the actual chemotherapy or the surgery or the radiation. It's everything else. It's all of the things that help kids keep well and healthy. And so one of the things that we have to work with very hard is that when we say, we'd like you to meet with a palliative care team, the first thing people do is take this deep breath and think, oh my God, my kid's dying and you won't even tell me. But that is in fact not the case anymore. And we've really evolved to having palliative care be a really critical part to us being able to treat the whole kid and the whole family. Are we managing pain correctly? Are we managing long-term side effects and how that kid is going to function when they return to the community, when they go back to school? Because we're really trying to manage all of the other parts of what we can do to help their lives be better. And that's an important thing. So I mentioned that because we need continued ambassadors in how we think about palliative care. It's not the treatment-specific care, it's everything else that goes into that. One of the privileges of having more and more kids be survivors of childhood cancer is they're going to grow up. They're going to need sometimes 30 and 40 and 70 and 80 years of care after their cancer diagnosis. So we have to treat adult physicians as well as we have to treat our own patients. We have to teach them what it is like to take care of survivors of childhood cancer. And we have to teach them what's normal and what we're worried about. We hang on to our patients as long as we can. My oldest patient at Children's right now is 33. My youngest patient at the university is 19, so there's a gray zone there. But it's actually a really important one because for a long time, internal medicine and family practice practitioners didn't have survivors of childhood cancer. They didn't know what that was. And so part of our job is to make kids healthy, productive adults. But we also have to help the people taking care of them know what that is. And so being able to evaluate lead effects, long-term side effects, and how to manage those sometimes for many, many years is really important. There's a whole field of outcomes research that we have the privilege of taking part in, and that's a great thing. And then I'm going to talk very quickly about immunotherapy because it's a big buzz right now and everybody wants to talk about it. But I'm going to show you this cool Star Wars picture just because I really love it and it's fun. And it comes from my colleagues and friends at the Children's Hospital of Philadelphia. This is, in essence, a way to make the human immune system fight cancer in an individual person. And I'm going to walk you through this just a little bit. T cells are the cells in our body that are responsible for helping us fight infection, viruses, a little bit of bacteria, fungus, viral infections like the flu, the common cold. T cells are critical for our immune response. There are other parts of the immune system that help T cells do that, and we'll forget Immunology 101 here, that's a whole separate day. But when we think about what the human immune system can do, it's actually smarter than any other therapy we can deliver if we can make it work right. And so what a number of scientists over the last 10 years or so have done is to learn how to take a human T cell, or lots of them actually, and engineer those T cells to be smarter. So what we've learned how to do is take these T cells and actually engineer them with this little cool looking fragment that's put into the T cells by a virus. It's introduced by a form of a virus, and it's a designer molecule, and we attach these pieces essentially to this that help the T cells stimulate not only other T cells, but their own growth and what we call persistence and their virulence, basically how much they can get angry. And we can engineer this part here, these sort of teal and green parts, to be targeted or fight against a particular antigen that's on the surface of a cancer cell. In this case, it's CD19, which is the most common antigen that's on the surface of ALL cells. So it's expressed on every ALL cell at very high levels. And if we engineer this green and sort of teal part to connect up to that thing that's sitting on the surface of the leukemia cells, what happens is it pulls those two cells in proximity to each other, and it allows the T cell to do what it's going to do naturally anyway. It's going to spew out its evil humors. It's going to make you feel like you have the flu and feel really bad, and it's going to kill the cell that it's connected to. So a bunch of really smart people have developed this technology. It's really exciting. The first sort of model disease was ALL, but it is not the only disease that people are working in. And this is an example. This is the only weird, geeky science picture I'm going to really show you. But this is an example that Dave Barrett and the group at Children's Hospital of Philadelphia have developed, where on day one, you take a series of lab mice and you give them leukemia. You inject leukemic cells into their tail veins. And by day 7, all of those mice have leukemia. That blue and green sort of intense yellow color is where the leukemic cells are. It's a phenomenal technology for being able to evaluate therapy. So by day 7, the 2 mice on the left are given these engineered T cells, and the 2 mice on the right are the controls. 3 days later, 72 hours later, the 2 mice on the left who got the engineered T cells are completely cured of their leukemia. And the 2 mice on the right are not going to do very well. Their leukemia has not only stayed there, but it's progressed. It's gotten hotter and redder, which is how we measure that. 72 hours. It's better than any chemotherapy that we have. It's more effective than any chemotherapy we have. It shows the power of harnessing the human immune system against a whole bunch of different diseases, ALL being the example given here. So what does that do when we do clinical trials? So if we look at about 60 children that at this point that had gotten this type of therapy, the expected survival for the children enrolled on this trial is less than 5%. And these numbers show that there's still a decline in the number of patients who are survivors, but starting about 9 months after the infusion, there are no further relapses, and about 50% of kids survive. The longest kid who is treated at the Children's Hospital of Philadelphia and has just celebrated her 5-year anniversary. That's a child who had the most refractory, worst, awfulest, aggressive, most horrible ALL, just like many of the kids who were enrolled on these trials. So these are durable remissions. There's no subsequent therapy for the vast majority of these kids. They get their T-cell infusion, and that's it. There's some kids that do go on to an additional bone marrow transplant or other therapy, but the majority of kids don't go on to other therapy. Pretty cool, pretty exciting, and pretty close to FDA approval. This is going to revolutionize how we treat childhood leukemia. So what happens? There are a whole bunch of trials going on right now. They are actually going on in a number of different kinds of disease states. And what we are doing is right now we are involved, there's a group of about a dozen of us designing the next generation of children's oncology group trials for leukemia. We're going to introduce these engineered cells called CAR T cells, or chimeric antigen receptor T cells, into patients at diagnosis, the patients that we know have the worst potential outcomes. And we're going to move that right up front and see if we can completely change their therapy. So that's very exciting. The other thing that's really cool is that we've now recruited probably one of the top 3 experts in the world who makes these cells, who will be starting at Children's in January, who's already on campus developing these clinical trials with us. We have a couple of trials already in place, and he will come and bring his team along with another of our recruits. Our new head of bone marrow transplant has been doing this with a slightly modified version of natural killer cells, not T cells. And so together, formidably, we have a pretty amazing set of armamentarium to do this with, and I'm very excited about that. It's another one where I feel like I should give up now because I'm not going to ever recruit anybody even more fantastic. But what's really cool is that we're developing these chimeric cells for bone tumors, for neuroblastoma, and for some CNS tumors. So it's not just for leukemia. And I think it's going to be really powerful. This is very new. This is all new for all of us. So their learning curve is steep, and it's very important that we do this well and do this right. I'm going to just show you a cool picture because I really like this and I made this slide and it makes me happy. But this is a different way of engaging the immune system, and it's with a set of monoclonal antibodies called T-cell engagers, just like we talked about bringing that T-cell in proximity to the tumor cell. This is a different way of doing it, a slightly different diagram. Philadelphia has much prettier graphics. I had to do my own. But what this is this little tiny piece of essentially a linker that pulls these two cells, the T cell and the tumor cell, in proximity to each other. And then it lets the T cell does what it's supposed to do anyway. The T cell comes in, it connects to the tumor cell, it spews out a whole bunch of bad stuff, and it's gone. This is a cool process because it doesn't depend on extracellular engineering. You don't have to make this in a lab. You just infuse this little linker in an IV bag, basically. You can do it independently of some things in the immune system that are a little bit more challenging. So this is an off-the-shelf product that doesn't have to be manufactured in a lab. It can be infused in clinic. Actually, we infuse it in the hospital for a variety of reasons, but it can do that in a bunch of ways that actually allow the immune system, again, to recognize the cancer, destroy those cells in the process. So this is the drug that I told you was approved in 2014, 2016. And it was done so in the same population of kids with ALL who have this thing called CD19 on the surface. And it's these engineered fragments that are pulled together. Once the T cell and the tumor cell are pulled together, the T cell just does what it's genetically and historically supposed to do. It's supposed to grow and divide and make a response against these other cells. And it does that, and in the process, it kills the leukemic cell. So very, very cool. Not a benign therapy. It makes you not feel very good if it's working. But again, very different from what we've thought about in terms of cancer therapy in the past. And again, every single one of these patients would have a less than 5% predicted chance of survival. And this drug induces very, very deep molecularly negative remissions in about 40 to 50% of patients. So very important statistically because these are patients that are saved with an infusion of a drug rather than more aggressive therapies. But I do think it's important as we think about how do we move drugs forward, what do we do to better therapy for the kids that we're taking care of? One is that we have to make scientific sense of this. What we're doing should biologically be reasonable. We also want to look at patients who don't have good options, that their chance for cure is poor, so that we're thinking about moving things up aggressively for them to give them a chance to benefit from these things. We also know that a lot of our therapy is intolerable. Can we do better? Can we give kids better therapy that is a little bit better tolerated, that has fewer side effects, both short and long term? And can we reduce the therapy for some, as we talked about? Can we think about substituting out those things to make lives better, to improve the quality of life, to improve the quality of the cure that we're seeking? And I'm going to sort of skip the commercial about how we do clinical trials more because you've been patient enough to listen to that. But I think what's also really important is that we make sure that we follow patients long enough. It's easy to follow patients for a few months or even a couple of years. But because many of these therapies are so new, we won't know for 20 or 30 years what the overall outcome for these patients is. So it's very, very important for us to be able to see what that is, to know how to make it better for the next kids who are getting treated. And so this long-term piece is really, really critical for us to be most thoughtful. I want to talk about this gap. Can we get to 100%? We're pretty close for many of our patients, and there are some disease groups that really effectively do have 100% cure rate. That's great. I want to focus on that gap and the place where the gap is the biggest, and so that's where a lot of our efforts and energy are targeted right now. So we're better at treating kids with cancer than we used to be. We're still not as good as we'd like to be. A lot of us would be very happy to be out of work. If the kids that come to our clinic every day didn't need us, we would be very thrilled to not have to go to work. But the only way that we're going to be able to do that is to continue to go back to the lab, continue to understand the science that's driving us, to continue to understand the tumors even when they make us feel really dumb. We need to get smarter. We need to collect that information and get better at what we're doing. So I'm going to finish with my friend Katie and her sisters. This is actually a pretty old picture right now. But it was the first summer that Katie went home after being treated here. But I always take a poll to see if anybody can guess which of these beautiful young women was our patient. You know what? It's interesting. Everybody always says the one on the left.
I think it's the one on the right.
It is. That's Katie. So Katie started high school this year. She was 3 years old when she came to us at Children's. She had just turned 3, and she was telling me, a very precocious 3-year-old, but she had lived most of her 3 years in the hospital. She was telling me that, and that was July 4th, that they lived out in the country, and that each of her older sisters had a dog. And so we were talking about dogs because I had a dog and they had dogs like my dog. And we kind of left it at that. And when Katie was home at Christmas, actually, Katie got home for Christmas that year, she called me on Christmas morning. I said, Dr. Gore? She had this little tiny voice. I said, hi, Katie, how are you? She said, I'm good, Santa came. I said, that's pretty special. And she said, Santa brought me a puppy. And so I kind of had to think about that for a little bit. And then her mom and dad got on the phone, and I said, all right, what are you guys getting yourselves into? And she said, Mom said, we've never gotten Katie a dog because we didn't think that she would live long enough. Every bit of it, she had never had a Christmas where she wasn't in the hospital. And this year we decided she could have a puppy. So that dog is now 13 years old. And I think has exhausted this family in every way. But Katie's in high school, and Katie's here not for anything special that we did, but because a lot of people took chances, and a lot of people were willing to try to move the needle when the odds were against them, and when we didn't have the answers, but we made guesses that fortunately paid off. So I will finish just with a quick acknowledgement slide, because the reason that Katie is here is very much because of a whole bunch of people who work long and hard every day, including Alex's Lemonade Stand and the Morgan Adams Foundation and my team at Children's that puts up with me, to really think about how we make these things better. Every day isn't perfect, but every day I think we're getting better. And it really takes gatherings like this and organizations like this and people like you to help us do that. So I do think I have the best job in the world. That sounds strange to many people, but I really think it's true. And I think it's a privilege to take care of the Katies of the world because they're making it better for all of us. So thank you for your time.